Greetings, everyone. My name is Chris Mathis. I'm the Chief Commercial Officer at AnaBios. It's my pleasure to moderate today's webinar sponsored by AnaBios. The topic today will be on the heart, which is one of the unique platforms at AnaBios. For those of you that haven't heard about our company, let me just tell you that we are a unique CRO and biotech company based in San Diego. AnaBios recovers human tissue samples from ethically consented donors, and we use the tissue or cells to perform physiological assays in which we can test preclinical compounds for drug discovery projects. We have essentially redefined first in human, and you can get more information about AnaBios at our website, anabios.com. And you might have noticed already in the GoToMeeting panel, towards the bottom, there's a handout section. You can download the two brochures related to AnaBios's unique cardiac platform. So it's my pleasure to introduce our speaker today. Dr. Tamer Mohamed is an assistant professor of medicine at the University of Louisville in the Division of Cardiovascular Medicine. Dr. Mohamed has a very interesting background. He did his PhD at the University of Manchester in cardiovascular and molecular medicine, postdoctoral fellowship at the University of Manchester, Gladstone Institutes. And in his bio, he says that his main interests are to identify novel therapies for heart failure, focusing on endogenous heart repair and regeneration mechanisms. As a pharmacist by training and the unique combination of academic and industry experience, his major aim is to perform translational research which directly benefits heart failure patients. He also has experience in the biotech industry when he was at the Gladstone Institute in the lab of Dr. Deepak Srivastava. He helped to establish Tenaya Therapeutics. And so he's back into academia. And we're really excited to hear his presentation today, which is entitled Human Heart Slices as the Next Generation of Cardiotoxicity Testing. So Dr. Mohamed, take it away.
Yep, thanks so much for the introduction. And it's my pleasure to give this talk today. So I think recently we have developed a system to culture human heart slices, and we believe this culture for human heart slices would have a potential to become a next generation cardiotoxicity testing platform, and of course efficacy for heart failure therapeutics as well, because this is probably our main interest to this. So just a background for this theory is that, so in drug discovery, as you can see from this figure, that the drug for any target or any potential therapeutic, if it's either a small molecule or gene therapy or whatever, it has to go through a pre-phase, which is the non-clinical testing, which is mainly done on vitro or in vivo or in silico, and then you file an IND, and then you go to clinical trial, and then surveillance after marketing as well. So the drug development cost and attrition become higher and higher once you reach to this stage of clinical trials. For the heart specifically, all of the models that we are so far using, we are using either in vitro, in vivo, or in silico models. I think our best bet so far is the human iPS-derived cardiomyocytes, which is human in nature, but is still lacking the 3D or the components of the heart, the full components of the heart. So that's why we usually use animal models to validate our targets, like large animals like pigs, monkeys, and so on, and dogs as well. And that's what's been in use so far in drug industry. But if you look to whether can we have a human in situ model, a human heart in situ model, the human heart is composed of many cells. It's not only cardiomyocytes. And many of these cells, like fibroblasts, monocytes, and microvasculature, we don't really still understand the nature of these cells. So there are different types. They are very heterogeneous, and the assembly of them within the heart is very complex as well. So to tackle this problem, we have recently developed a physiological biomimetic culture system for big and human heart slices. To be able to culture human heart slices or big heart slices for at least 6 days. This is a reasonable time to be able to test drugs efficacy or toxicity, at least for the acute and subacute effects. So the way that we do this is we get a human or a big heart, then we get blocks of it, and then we slice it with a vibrotome to a 300 micron slices. And then we apply, we optimize the media, and we apply continuous electrical stimulation to them. And we apply continuous oxygenation, as well as we change the media 3 times a day with freshly oxygenated media. And with these conditions, we've been able to reach a 6-day culture where the heart tissue keep intact and functionally similar to the fresh heart tissue. So what can we do with this? We can do lots of assays. So we can do calcium imaging, as you can see here. We can isolate cardiomyocytes, and we can do electrophysiology on them. We could do contractile function measurement, either using an organ path to measure the contractile function. And we recently adapted this high throughput contractile measurement using impedance technology with this device called Cardiotoxicity from Nanine. And we can measure metabolic flux on these heart slices. We can do transcycloptomics. We can do electron microscopy. We can do immunofluorescence for whatever. We can do Western blotting as well. And we have collaborated with Dr. Tim McKenzie in Colorado as well to do a single myofibril contractile function on these heart slices as well, as you can see. So all of these are published data. You can go through the publication as well. I'm going to go through a few of them in this presentation. So just to show you how to do that. So slicing, so how to slice and how to culture. We made it pretty simple, so everybody and everyone can use it and hopefully adapt it in his lab. And we have published a video protocol for this in Jovi early last year. And just to give you, an essence of how we do that. So we usually start with perfusion in situ in the big. So this is a big. I couldn't image the human, but for the humans, we process it the same way as the pig. Sorry. Oops. Sorry for this technical issue. Okay, so we do in situ perfusion at first with cardioplegic solution to slow down the heart, and then we take the heart out and perfuse it with more cardioplegic solution just to stop the movement, and then we dissect the left ventricle, as you can see here, into strips like this. And then we cut it into about 1 centimeter to 2 centimeter cubes to be able to slice them. Okay, like this here. And then we stick them into this holder on an agar piece. So we stick the agar piece first on the vibrotome holder, and then we stick the tissue on it. And this glue is the polyacryl glue. So it's whatever you use to do stitches. If you have a cut in your hand or something like this. So it's a basically human tissue compatible glue. And then this is the holder for the vibrotome. So we are using vibrating microtome to be able to slice. And as you can see here, this is the blade is vibrating while it's slicing, and we are providing oxygenation in the path as well. As you can see, the bubbles are coming out. And then once the slice is done, you can take it and you can transfer it into into a thyroid solution where we leave it for some time with the thyroid, because this has got PDM in it, so it stops the contraction of the heart. And then we start trimming the slice, as you can see here. So we make the slice flat and then we trim it. So we get rid of the edges, of the cut edges, because these edges basically deteriorate the life of the slice afterwards. So I'm just speeding it up a bit. So yeah, so you trim the edges. And then after trimming, we stick a polyurethane, pieces, strips to it, for the slice to keep attraction from the two ends, and we keep it away from the bottom of the plate as well. So they can basically contract while they are in the culture, and freely contract while they are in the culture. So, sorry. All right. So then after we stick these two from both ends, so you stick these two to the two edges of the slice. And then you place the slice into the culture plate. So usually we do it in a 6-well plate. Okay. And after you place it, we place, oh my God, I'm sorry for that. So yeah, so then we place it, and then we place the cover of for the electrical stimulation. So we have a C-Pace. So the C-Pace is commercially available system from Ion Optics, and any lab can buy it, and you can just place the top, the electrical stimulation top, and you just need to connect it to you need to connect it to the C-Pace stimulator and program the stimulation. So we usually use 10 volts and 1.2 hertz for the stimulation, as you can see here. So here you connect it to the C-Pace stimulator, and this will provide continuous electrical stimulation to the patient. So, okay, so this is so why did we get into this? So I think this is a pretty interesting story, because this is the serendipity of science, basically. So in October 2016, we started Tenaya Therapeutics, which is a biotech company that's developing new therapies for heart failure based on cardiac deprogramming approach and other gene therapy approaches. And this company, I've been the first scientist in it, and then and now it's growing. It's about 100 people now, and they just raised 100 million to take the directive programming into clinical trial. When we started in the company, we wanted a human system to test our gene therapy on it. And how can we get human heart tissue in culture to be able to test the efficacy of our gene therapies? So at this time, we've been developing cardiac reprogramming, as well as we have found a way as well to induce cardiomyocyte proliferation by inducing by infecting the cardiomyocyte with 4 cell cycle factors: CDK4 cyclin D and CDK1 cyclin B. And this was published 2 years later. Anyway, so we wanted really to have a system for this, and what with the what we found is the human heart slice system would be really the best thing to do. And we actually did the experiment, and if you look to our paper in 2019, we have been able to induce the cell cycle in these human heart slices with these 4 cell cycle factors, as you can see in this figure. But anyway, this is not really the topic of this talk. But how did we? So when we in October 2016 and early 2017, the status of the heart slice technology was not really as good as it is now. And I'm just gonna show you how the status of the field is. And so there were 3 major groups that were working on this, and one of them is Dendorfer group in Munich, where they have first published a human heart slice culture on Transwell in 2012, but this heart slice culture doesn't really keep the heart slices viable for too long, because after only 1 day the contractility drops by 90% almost. So then Dr. Efimov in George Washington University has developed a similar system where they culture them on Transwells as well. And he's been able to do optical mapping on human hearts for 24 hours as well. And then Dr. Terziano group in London in the Imperial College, they have as well developed a new system, which is they can submerge the heart slices while they stretch it a bit. And this kept the heart slices pretty viable for 2 or 3 days, which was interesting and exciting. So because I've done my PhD in Manchester and I know the Imperial College very well, so we talked to Dr. Terziano and we and he's been very generous to allow us to collaborate with him, and we sent our technician over there to London for 2 weeks to learn how to slice hearts and to learn from him his recent advances in the technology and so on. And we asked him, we wanted, a simple, reproducible, medium throughput culture system, which can fully preserve the heart slices viability and functionality for at least 5 or 6 days. So we will be able to infect our viruses and look to the effect of them. And he's been, very generous and allowed us to learn how to do this technology and work with him to develop it even more. And so what we've done after being learning from him and from his lab is we have tried to induce first electrical stimulation to the culture system, because we thought, okay, so these transwells, they are not really electrically stimulated or anything, we just leave the heart as it is. So really the heart is usually electrically stimulated all the time. So we've done the first introduction of the electrical stimulation, but this was very disappointing, because with the normal media that's been in use for the heart slices, which is consistent of medium 199 on ITS, with electrical stimulation, we've seen a major drop in viability after only 1 day of culture. But so then we went back and said, okay, maybe this with electrical stimulation, we are imposing, the heart slice needs more nutrients and so on to be able to compete with the electrical stimulation. So we have screened various conditions, and we reached out the conclusion that we have to add at least 10% FPS and FGF and VGF to the culture media to be able to maintain the hard slices viability. And this, as you can see here, with these components, with these new components, you can keep them up to 6 days with full viability. And structurally, you can see as well here from the troponin and the connexin staining, you can see with these for 6 days, we see the same connexin and troponin structures that we see in day 0 or the fresh heart tissue, compared to the transwell culture, which really they deteriorate their troponin and connexin as well. And functionally, we've seen as well that at day 6, we have similar calcium transients as day 0, as well as response to isoproterenol. But after day 6, really, the functionality and the calcium transients goes really haphazard into these culture. And then the contractile function as well have been maintained for 6 days, similar to day 0. And what's really interesting to look globally to the gene expression, we've done RNA sequencing. And after 6 days, we have only 5 genes that have been misregulated between comparing day 0 and day 6. But at day 10, we have found a lot of genes that have been either up or down regulated. And we knew that, so if you look to these genes that at day 10 they have been down or upregulated, you will see that most of the cardiac genes have been downregulated, and most of the inflammation and fibrotic genes are upregulated, which means that the heart slices after 6 days they go into a bit of dedifferentiation mode. So we were not the only group that have shown advancement in this technology. Dr. Tarciano group at very similar time to our publication have shown as well that addition of preload to the heart slices by stretching them at a 2.2 sarcomeric length. This actually they had heart slices viable for at least 5 days. And Dr. Dendrofer group in Munich, they have developed this engineered system where they applied preload and afterload to the heart slices, and they have seen maintenance of contractile function of human heart slices for up to 16 weeks. And this is really interesting and exciting. And Dr. Efimov group in Washington as well, they have done a really cool bioengineered device, like a heart-on-a-chip device, where they can keep the human heart slices viable for 3 days on this heart-on-a-chip. So prolonging this viability of the heart slice actually, and this all happened in 2019. So this really made this technology amenable for many applications. And the simplest first application would be to predict cardiotoxicity and to be reliably predicting cardiotoxicity. So I've been working with Dr. Efimov and George Washington University and Dr. Ribeiro and the FDA on this question, and to know whether the heart slice really can reliably predict cardiotoxicity compared to the system that's available now, which is human iPS-derived cardiomyocytes. And we just recently published this a few months ago in the Toxicology and Applied Pharmacology journal. So as you know, that cancer or anti-cancer drugs, many of these categories have a lot of cardiovascular impact. So they have a lot of cardiotoxicity. And to be able so this was really the first study to do on this. So we choose 3 different categories of cardiotoxins that could that have different mechanisms in inducing cardiotoxicity. So, and we asked the question whether would the heart slices predict cardiotoxicity or the correct mechanism of cardiotoxicity of these drugs compared to the human iPS cardiomyocytes or not. And so to do that, we choose 3 categories. Doxorubicin, of course, everybody knows doxorubicin. It's inducing a lot of oxidative stress on the cardiomyocytes, so it has got a direct effect on the cardiomyocytes. We tried Herceptin or trastuzumab, and the trastuzumab basically is affecting directly as well the cardiomyocytes. And the other category is sunitinib. So sunitinib doesn't really have too much effect on the direct effect on the cardiomyocytes, but it affects more the microvasculature. And this is pretty known in clinic. And that's why the iPS cardiomyocytes is really not the best model to detect its cardiotoxicity. So we started with these data from Dr. Ribeiro lab in the FDA, where they have run some tests on human iPS-derived cardiomyocytes. And they have used aspirin as a control, so you can see there is no toxicity. So this measure is the caspase flu assay. So it's basically apoptosis assay. And so there's really, so the aspirin, of course, doesn't induce at any dosage, doesn't induce any toxicity for human iPS-derived cardiomyocytes. But doxorubicin, as low as 0.5 micromolar, it's inducing cardiotoxicity, as you can see here. But sunitinib, at low dose of 1 micromolar, it doesn't really induce anything, any cardiotoxicity. But at 10 and 60 micromolar, it really kills the cells. But if you look to the serum level of sunitinib, it's within this range. So basically at this range, the sunitinib doesn't have any major effect on the human iPS-derived cardiomyocytes. So we designed our experiments based on these data. So we have done a 48-hour treatment. We've treated with doxorubicin. We used 3 different dosages. Herceptin, we used 3 different dosages, and sunitinib, we used 3 different dosages as well. And the lowest dose is basically the clinical dose. So I'm gonna focus here on the clinical dose, and you can check the publication for the other dosages, which is more exaggerated phenotype anyway. And then we assess the calcium transients, we assess contractility, connexin 43, and troponin as a structural phenotype, as well as the whole transcriptome to look at the mechanism as well. So as you can see here from these pictures, so this is the control heart slice. It's structurally very well organized. The connexin is on the edges of the cells and so on. With doxorubicin, with the lowest dose of doxorubicin, we get really a big disruption of connexin 43, as you can see here. With trastuzumab or the Herceptin, we have really a big damage in the cardiomyocyte. They even don't stain with troponin anymore. So there's a really big damage for this. And for the sunitinib, there's really not much structural damage at this low dose as well. So the connexin, as you can see here, the connexin is aligned on the edges of the cells, and it's nicely aligned. So structurally, there's not really a major difference, and might be this what we see on the human iPS cardiomyocytes. But functionally, as you can see here from these calcium movies, so this is a flow for that loaded heart slices, and this is with the control slice. So you can see the calcium transients are nice. With the doxorubicin, we don't see any even loading of calcium because it's just, you know, all the cardiomyocytes probably are not functional anymore. And with the Herceptin, we see a lot of irregular calcium movements. So there's a lot of arrhythmia. Probably this was due to the damage for the cardiomyocytes. With the sunitinib, actually, functionally, we see a lot of overloading of calcium, as you can see here, and a very slow on the movement, slowing in the movement as well of the calcium. So this is a functional effect, and now the heart slice doesn't move anymore. You see this one is moving and contracting, but these ones are not moving anymore, and this probably because of this calcium overload phenotype. Oops, sorry. So then we have used the cardioexcite to assess the contractile function, and we've seen only the control has got really nice contractile function compared to the other three drugs. If you add any of these drugs, the contractile function is completely inhibited. And we see this as well in the calcium movies that I showed earlier in the earlier slide. So what about the mechanism then? So is the mechanism in these heart slices is similar to what we see in the clinic? So doxorubicin, for example, is known to induce oxidative stress. So can we see this? So in this, the RNA sequencing comparison between the doxorubicin and the control showed few hundred genes up and few hundred genes down. And if you look to the genes that are down-regulated, so they are mostly focused on the heart genes as well as the cell division genes, because we know that the doxorubicin is affecting directly the DNA synthesis and the nuclei because of the oxidative stress that's inducing. And if you look at the upregulated genes, the inflammatory and oxidative stress comes the first go term as well. So this means that, you know, we are seeing very similar mechanism for whatever known as on doxorubicin. So for trastuzumab or Herceptin, it's known as well to its direct effect on the cardiomyocytes. So we have few genes are down and few hundred genes are down, few hundred genes are upregulated. And as you can see here from the go terms, the cardiac muscle contraction genes are coming as the first hits on these downregulated genes, which means there's actual cardiomyocyte damage. And there's a lot of upregulation in the translation and cell division, which we don't understand too much about this, but I think this would be interesting to perform new studies to understand why the Herceptin is increasing the mitotic spindle organization and so on, and translation especially. For the sunitinib, the sunitinib is known to affect the microvasculature more than the contractile proteins. So for the genes that there's few hundred genes are up as well, and 50 genes are up and 180 genes are down regulated. And if you look to the down regulated genes, they are mostly focused on either blood vessel or angiogenesis go terms here, which means that it's predicting what actually the sunitinib is doing, which is affecting the microvasculature. And if you look, the upregulated genes is mainly the immune response, because probably it's inducing some inflammation as well. So to conclude this, I hope I just show you that some data to show that the heart slice culture system has got the potential to be considered as a secondary screen to predict human acute and subacute cardiotoxicity, to close the gap between preclinical and clinical testing results. So we can actually, whatever fail, you know, whatever give a false negative in the human iPS cardiomyocytes, we can screen it on these heart slice and get sure that it doesn't really induce high cardiotoxicity because it passed the human iPS cardiomyocytes. What are we doing now? We just recently joined the Health and Environment Science Institute, and there's a cardiac safety committee within this institute. So this institute is including a lot of biotechs and large pharma, as well as the FDA and many regulatory bodies over the globe. So it's not only the American part. So there's people in Europe and Canada and Asia as well. And on this cardiac safety committee, this is basically phase 2 of the CEPA, if anybody knows about the CEPA. So in this cardiac safety committee, we have 13 known cardiotoxins, where many labs will be testing them on human iPS-derived cardiomyocytes. And we are gonna do a parallel testing on human heart slices as well. So we have a comparison of 13 major cardiotoxins from different categories. So this will work as a really good validation for this technology in terms of cardiotoxicity testing. So what are the remaining challenges for this technology? I think one major challenge is the availability and accessibility to fresh human heart tissue. And we are privileged to have access to these through AnaBios. And AnaBios as well has got direct access to the fresh human heart tissue. So I think this is really an advantage. For the academic labs, I believe you don't really have to do on human heart tissue. If you want to do pig or dog or cat, you can do anything from rat and above. So we can't do mouse because it's too small to slice. But from rat, we can do heart slices on rats, and many people have done it on rats as well. The other point is we need to emulate the full cardiac cycle in the culture to be able to extend it longer than 6 days. Like the work done by Dendrufer, actually, they have extended it for 4 months. Even though this is not the full cardiac cycle emulation, but at least they have emulated most of the cardiac cycle. Further optimization for the culture media as well is essential, because probably we need more nutrients to provide to the heart slice, more than what we are doing now, and this might keep them happier in culture. And the other thing is, if we can produce a continuous monitoring of heart slice functionality within the culture, this would be great. So if we can monitor the electrophysiological properties as well as contractile function, as well as metabolic function within the culture, this would be really great, because so far, all the assays that we do, we take the heart slice after we culture it for a certain amount of time, we take it and do the assays outside. But doing it in the culture will allow us to do a lot more and do a lot more understanding as well. At the end, I would like to thank my group, who's done really all the hard work, and our collaborators around the globe as well. And I would like to thank the American Heart Association, NIH, and the Department of Defense for funding. And I'm happy to take any questions.
Thanks, Tamer, for excellent presentation. We do have time for questions. So if you look at the panel, there's a question box, or you could simply raise your hand and ask verbally. I think I can get things started. So, Tamer, excellent presentation. You have a picture here of isolated cardiomyocytes. I'm wondering have you done any systematic comparison between the slices and the isolated cardiomyocytes, whether pharmacologically or with gene expression?
Yeah, so we isolate the adult cardiomyocytes from the heart slices. So we just treat them with collagenase and isolate them. We haven't done, and this basically Dr. John Satin in University of Kentucky has done a lot of electrophysiology measurements on the isolated cardiomyocytes from these heart slices, but we haven't done really a systematic comparison of the electrophysiology that's measured from the heart slice and from the isolated cardiomyocytes. But I think this will be really interesting. So now we are trying to incorporate multi-electrode array measurements on these heart slices. And hopefully we can be able to compare from the same heart slice the electrophysiological properties between the isolated cardiomyocytes and the heart slice.
Okay, thanks. We are getting some questions in from the audience. I'll go to a couple of hands that are raised first. Aravind Rahman, I'm going to unmute you. Go ahead and ask your question.
Yes. So it was a good presentation. I have a question based on how you measure the contractile force. Is it a relative measurement, or is there do you follow any other technique to measure it directly?
So yeah, so we have two ways to measure contractile force. and one is with the organ path, as you can see here in this slide. And the other one is using impedance technology with the Cardiotoxicity. So does this answer your question?
Yes. Thank you.
Yeah.
Okay. Another question from Carlos Albuera Paz in the audience. Go ahead, Carlos.
Yes, thank you, Chris. Nice presentation. My question is how you implemented the tissues in the Cardiotoxicity. This is the first question. And the second, sunitinib is a heart channel blocker. Did you see arrhythmias with sunitinib?
Sorry, can you repeat the first part again?
The first question is how you implemented the tissues, the tissues in the Cardiotoxicity. So you add just a piece of the tissue in each well?
Oh yeah, so the Cardiotoxicity is a 96 well, and we have punches which is the size of the 96 well. So we punch the tissue. So it's I think it's 6 millimeter punches, and we put the slice on the Cardiotoxicity, and then when the Cardiotoxicity has got stimulator plates, so it stimulates the heart slice, so it contracts and it measures the movement during the contraction.
How you attach the tissue on the wells? Did you use this blue glue?
So the
how you attach the tissue to the electrode?
Well, the tissue lay on the electrode, so the bottom of the plate in the Cardiotoxicity is actually electrodes.
Yes. No, I am familiar with
it on this, so the electrodes will sense the tissue.
Okay. And the second question was if sunitinib is a heart channel blocker. Did you see arrhythmias with sunitinib at those concentrations?
We haven't assessed arrhythmias. We are now trying to implement a multi-electrode array system that we are collaborating with a new company called Mapping Lab to incorporate their system in our heart slice system to be able to assess arrhythmias, but we haven't assessed electrophysiology during these experiments. But this is the plan for the compounds from the HESI as well, from the Cardiac Safety Committee from the HESI, the 13 compound that we are going to test.
Thank you.
So a couple of questions coming in from the panel. Here's one. Can you cryopreserve the slices?
It's a big challenge, yeah. This is really a big challenge. I think we've tried it. Dendorfer group in Germany, they have done that. I'm not sure how they done it, but they describe it in the paper. We've tried it and we haven't been successful in doing it.
Okay. Just a reminder, there is time for questions. You can use the panel in the lower section. There's also a couple of handouts from AnaBios that you can download. Another question from the audience and comment, terrific work. How do you protect against reperfusion injury? And secondly, how many slices can be created before viability of slices tails off?
So to protect from reperfusion injury, we are trying to be as fast and quick when we are doing the slices. When we've done the RNA sequencing and we didn't see really not much, there's no much changes that I just showed you up to 6 days from day 0. And what we do as well to protect from this is we perfuse the heart with 2 liters of cardioplegic solution in situ and in vitro. So it's fully perfused before we do the culture. So can you repeat the second question again? I forgot it.
How many slices can be created before viability tails off?
Yeah, so we have really a big high production on this. So we have 2 vibratomes working simultaneously, and usually we operate between 72 to 96 slices from the heart, from each heart, and we can do this in 3-4 hours.
Okay.
So the operation
And,
is around
yeah.
100 slices from each heart.
Okay, thank you. You know, AnaBios is very interested in this technology, and we see this really as the next frontier in our cardiac platform. And what separates us really is our high quality and the extensive network of hospitals and OPOs around the U.S. It was interesting to hear you mentioned extending the days in culture and emulating the cardiac cycle. What do you think are the difficulties in achieving that? What are the challenges there?
So there are we are working actually on this emulation of the cardiac cycle, and I believe probably many other labs are working on it. We are almost there, I believe, on doing this, and this should extend the cardiac, the heart slice viability even much longer. I believe maybe for a month or two, I don't know, but we will, if we can do it like, correctly, and how the heart is, I believe this will be really great. But so far with the 6 days culture, we are good for at least acute toxicity, but I think to keep it for a very long time, I think this will be really exciting.
Here's another question from the audience. Do you need to use fibronectin or other coating in order to adhere the slices on the cardioxide plates? I think this comes back to Carlos's questions about how do you get the slices.
Yeah, we've tried different. Yeah, so for the cardioxide, we've tried different ways of optimization for it. We've tried to coat the plates. We've tried to put some weight on the heart slices. But actually, this didn't make any difference to the recording. So the recording is very nice just from laying the slice down to the electrode. You don't really need anything to stick it because they contract and go back. They contract and go back. So they are very nice. The thing is, you can't do too long recording. You just need to record, for 5-10 minutes. After this, the slice gets exhausted from the too much workload that you are imposing on the cardioxide. But it's you don't need anything really.
Yep. Okay. Yeah. Can you remind me how thick is the slice?
It's 300 micron.
Yeah, I suppose that that's big enough and heavy enough that it really just sets down on the electrodes.
Yeah, it's heavy enough to just sit down on the electrodes, and the electrodes does the job basically.
Here's another question from the audience, and it relates to hearts from diseased patients. Have you been able to use slices from HCM and DCM donors? Yeah,
so the only thing that we tried is from infarct heart. So we have received few hearts from after myocardial infarction, so they died basically from infarction, and we have been able to slice the border zone as well as the remote zone, and we've been able to keep the heart slices from the border zone very similar time, and this was included in our publication as well in our safety research paper.
Okay. And related to that, with those preparations, how long can you record? Is it possible to record 3 to 6 days also, or are they too damaged for that?
To record what? To record?
From those preparations from the diseased hearts.
Yeah,
how long
so
can you record
we've been able to record everything for 6 days.
Okay, interesting.
Yeah.
All right. Have you been able to look at the cardiac fibroblasts in situ?
Yes, we have collaborated with a lab that's interested in fibroblast, and basically you can stain the fibroblast with periostin or with alpha SMA and see the expression. There's another lab as well was interested in looking to the endothelial microvasculature, and you can stain as well and look at them. And when you collagenase, when you do the collagenase digestion as well from them, you can as well get the cardiomyocytes as well the fibroblast subpopulation, and then you can sort the fibroblast with Phi1 or something like this. But we've been collaborating with a few people that are interested in the other subtypes, and you can get whatever you want from them, and it's easier to get it from the heart slicer than getting it from the whole cube of tissue eventually.
Right. Okay. Well, Tamer, thanks again for an excellent presentation. I know that Naja and the team at AnaBios is really excited about collaborating with you on this project. I'd also like to thank my colleague Gary Watkins for putting this together, and we look forward to seeing all of you at a future AnaBios webinar. Thanks again, Tamer, and goodbye, everyone.
All right. Thank you so much. Thank you. Bye.